All right, so this is part 3 of your outline today on chapter 2 for anatomy. That we're going to go through all the organelles inside a eukaryotic cell. So this is starting from letter C in your outline. Number 1 is the cytosol. So the cytosol is the internal contents of the cell. So beneath the membrane, beneath the cell membrane or plasma membrane, you have a jelly-like fluid that's inside. It's different in ion concentration and molecular makeup than the outside. And it's what houses a lot of the chemical reactions that you have. There's a lot of chemical reactions going on inside the cytosol. And you may have also heard the word cytoplasm. Sometimes the word cytoplasm and cytosol are used interchangeably. And so we're looking at basically the yellow area here that would represent the cytosol. So let's talk about the organelles inside the cytosol. So the organelles are listed for you here in letter B, so we're going to go through each one. The first organelle here is the nucleus. So the nucleus of your cell is going to contain your genome. The word genome is for all the DNA inside your cell. And I should sort of clarify that the genome inside your cell, you do have most of your DNA inside your nucleus, but you also have some DNA inside the mitochondria. It's called mitochondrial DNA. So the nucleus will have all of your chromosomes that are required for life. And you can see that the membrane here has pores in it. There are openings to this nucleus, and that's to allow rRNA and different kinds of transcription factors. There's different molecules that need to interact with your DNA. So there are nuclear pores, regular pores within the nuclear membrane. The nuclear membrane is actually a double membrane structure, so that's doubled up. And inside, we have the material inside. The cytoplasm inside is actually called the nucleoplasm. So they have a different name for the environment inside the nucleus. And then you have a structure called the nucleolus, right here, the nucleolus. So this is a really dark dot inside the nucleus. If you see this picture here, we have our large nucleus itself and then a dark nucleolus. What goes on there is making ribosomal RNA. Okay, so that makes something called little r RNA, ribosomal RNA. And this ribosomal RNA is used to create ribosomes. So that little r is for ribosome. So the nucleolus has a job to make something, right? And all the rest of this substance that you see that's dark is called chromatin. So chromatin is a form of DNA. DNA has 2 forms to it. You should remember this from Bio 3. The looser kind of form that's drawn a little bit like spaghetti, that is chromatin. So chromatin is the usual form that DNA is in. But when your cell is reproducing, that chromatin gets tightly packed, and those tightly packed units are chromosomes. So this might look familiar to you, right? So when the cell is dividing through mitosis or meiosis, it becomes a chromosome. So the word chromosome is used 2 ways. It's to describe each strand of DNA, chromosome 1, chromosome 2, but it's also used to describe this discrete packet, this wound up, tightly coiled DNA. Okay, so that's our nucleus. Let's move on to the next organelle. Oh, this is a nice picture of your cheek cells. Okay, so that you can see that if you scraped your cheek, this is what you'd see. This outer edge of a cheek cell, of course, is the cell membrane or plasma membrane. All the light pink area here, right where my X is, that's the cytoplasm or cytosol. You can see that name here. And then the dark spot in the middle, that's your nucleus, okay? So those are the 3 things that you should be able to label if you see a picture like this. You should be able to label the cell membrane, the nucleus, and the cytoplasm. All right, so our next organelle is the ribosome. The ribosome is a organelle with 2 subunits. So they're both proteins, there's a smaller subunit and a larger subunit. A lot of times ribosomes are drawn kind of like this, just a smaller subunit on top. And ribosomes make proteins, right? So their job is to help synthesize proteins. Without them, you could not make proteins. So that's all you need to know about the ribosome. The ribosome is made of two, I do have in your notes that's made of ribosomal RNA, as we discussed. Ribosomal RNA is made in the nucleolus plus protein, right? And that's what a ribosome is. All right, let's move on to the next one. This is the endoplasmic reticulum. There are two kinds of endoplasmic reticulum. There's smooth and there's rough. Your notes begin with a rough ER, so rough endoplasmic reticulum is rough ER. And you can see the roughness is due to ribosomes that are attached to this membrane. Now, if you look carefully at what's going on in this picture, the nuclear envelope is, I don't know if it's actually drawn in, I think maybe right here. The nuclear envelope becomes the ER. So it's the same membrane that just becomes folded and convoluted in this ER form. Some of that membrane is dotted with ribosomes, and then we call that rough ER. Because ribosomes are on it, the job of rough ER is to make proteins, okay? Because ribosomes are there. But the proteins that are made on the rough ER are usually fated to leave the cell. So these are going to be called secretory proteins or secreted proteins, right? They're secreted from the cell. Ribosomes are also present in the cytoplasm. So I should mention that those ribosomes in the cytoplasm, they are called free ribosomes. And the proteins made there usually stay inside the cell. Okay? So either way, if a ribosome is present, proteins are being made. The other kind of ER is smooth ER, which we can see here, right? So this is rough ER because it's rough. This is smooth ER because it's smooth. And it's free of ribosomes, so there's no ribosomes on it. And the reason is because there's no protein making going on here. The smooth ER is to make lipids. So it actually does make products. It makes lipids. Let's review what kind of lipids might be made here. Phospholipids, right? Phospholipids are in constant need because your cell, all of your membranes are made of phospholipids. It can make something called cholesterol, okay, which is a lipid. So those are two good examples of lipids that are made there. The other job that the smooth ER has is to detoxify. So a detoxify your cells. What that really means is if you have a molecule that is something that your body wants to get rid of, it will modify that molecule to change it slightly so it's not so harmful, and then also help your body get rid of it through your urine. So that is your smooth ER. Okay, so I have a question for you in the outline. Which organs' cells would have a large amount of smooth ER? So what organ do you think has a big job of detoxification? And if you said liver, that's a really good guess. That's correct. So the liver's job is to detoxify substances in your bloodstream. And so the liver cells will have a lot of smooth ER because that's a predominant job the liver has. All right. So here is the overview, right? Smooth ER for lipid metabolism and detoxification. Rough ER, making proteins to be secreted from the cell. This is just a closer look at how the rough ER makes proteins. So here's our ribosome. Our ribosome attaches to the ER. This is the membrane of the ER. And so the ribosome will come in, and what's being dropped inside the ribosome right here is a protein. So this is a new protein that's being made. And then the protein will get shipped away out of the cell because the ER will form a vesicle. So you see how it's budding off, and then it's going to pinch all the way off. It's going to pinch off into a vesicle, and this one's going to go to the cell membrane to get secreted. So for exocytosis, phagocytosis. Okay? All right, so that's our ER. The next organelle we have is the Golgi apparatus. The Golgi apparatus is also sometimes called Golgi body, so you may have learned Golgi body in Bio 3. So the Golgi apparatus is a series of flattened sacs, 3 to 5 flattened sacs, but the sacs are hollow. So if you take a look at what's going on here, you have, I like to say pita bread because it's this oval, hollow membrane, and it's stacked, something like this. And there is a in face, so you can see how the arrows are pointing in. And then on the other side, it is an out. So the Golgi apparatus accepts molecules on one side and then secretes them on the next side. When they accept the molecules are traveling from the ER. So here is our endoplasmic reticulum. We can see that we have ribosomes here, so this is a rough ER. We can see that proteins are being shipped from the ER to the Golgi apparatus. The Golgi apparatus then accepts them. You see how the vesicle fuses with the Golgi apparatus in this image here and also in this image here. And then the newly made protein inside the Golgi will get modified. So if there's something that needs to change about the protein, if there needs to be an oligosaccharide attached to the protein, to make to make a glycoprotein, that's going to happen in the Golgi apparatus. So it sorts, it processes, it packs and ships the material via these vesicles, and then after it's done modifying the materials inside, it'll ship it out to where it's supposed to go. Okay, so the other thing about the Golgi apparatus is that lysosomes are created here. Okay, so let's take a look at this. So this is a lysosome. A lysosome, just moving on to the next organelle, a lysosome has a acidic interior, right? So inside the lysosome is about a pH of 5, which is acidic. It will also have digestive enzymes. Okay, and so this is basically a very digestive organelle. When it fuses with a vesicle that has something inside of it, it'll digest whatever's inside the vesicle. So this is something that's created in the Golgi apparatus. All right, so let's take a look at, so I just want to mention too that lysosomes, under lysosomes 3, autophagy means to self-eat. And that means that if there's any organelles that the cell does not require, or it's broken or not functional, it can actually digest its own organelles and use the parts to recycle, right? This is a nice image of the job of lysosomes. So here's the word autophagy. So look at what's going on in autophagy. So let's just start here. So we have the Golgi apparatus. We have a lysosome being created from the Golgi apparatus. The lysosome is breaking down. This is, if you recognize this, this is a little piece of the mitochondria. So the cell doesn't want this mitochondria anymore. And the lysosome comes in, it digests it because all those dots represent digestive enzymes. And then this is now considered either waste, right, and can be exocytosed, or it could be recycled and used inside the cell as nutrients. The other image and drawing you have here is the cell taking in some kind of food. So remember that if you have a solid substance coming in, it's called phagocytosis. You can see this invagination of the cell membrane. Now we have a food vacuole, and we need those lysosomes to fuse with the food vacuole, release its enzymes inside, and you get digestion. Okay, so lysosomes are for digestion, in short. Moving on to the mitochondria, we have mitochondrial DNA first. It is a double-membraned organelle with DNA inside. So remember the mitochondria also has its own DNA. So it kind of acts as a autonomous organelle. It has the capacity to make more of itself. And if you remember from Bio 3, the mitochondria was thought to be this parasite a long time ago. And after millennia, that parasite lost its ability to live on its own, But the parasite, of course, at one time was a free-living individual and had its own DNA. So we think that the mitochondrial DNA is left over from that parasite. And that's why it has its own DNA. It's double membraned, and you have to remember that this is where ATP is made. So ATP, which is right here, right? We see this. This is what cellular respiration does. So remember, we're not going to go over cellular respiration, but cell respiration is where you take glucose, you produce ATP with glucose, and this is your energy of the cell. So how many mitochondria a cell has is going to be an indicator of how active that cell is. So you can see here, liver and muscle cells can have as many as 300 mitochondria per cell. So if you have a very active if you have a very active cell that needs a lot of energy, it's going to just have more mitochondria. If you have a more quiescent cell, a cell that's not very active metabolically, it just doesn't need that many mitochondria. So it might have like 100 mitochondria or might have 50 mitochondria. So it just depends. All right. Peroxisomes. It looks like I don't have a picture of peroxisomes, but peroxisomes are small, tiny. They look like lysosomes, but they're smaller, and they're going to have oxidases that neutralize free radicals. And I have in the notes that free radicals are really harmful to humans. They can cause damage to the DNA. So when you have antioxidants, when you eat antioxidants, that will clean up your free radicals, and peroxisomes will help do that as well. Peroxisomes help to detoxify alcohol, formaldehyde, phenol, other harmful compounds, and they also help to catabolize fats. And I have a question of what organs do you think would have a lot of peroxisomes? And again, the liver, because the liver's job is to detoxify. Moving on to the cytoskeleton. So for the cytoskeleton, it's a network of fibers. These fibers are made of proteins, and there are 3 kinds of fibers that are all considered cytoskeleton. They are the microtubules, the microfilaments, and the intermediate filaments. The cytoskeleton has a lot of sort of different jobs inside the cell. So it will maintain the cell's shape. It acts like a scaffolding inside the cell, so it's like a meshwork. It also is dynamic, so it's not like your skeleton that you think of as like your bones are just set, they don't change. This cytoskeleton can get longer, get shorter. It's very dynamic. It can be built in different ways. So it helps the cell itself move around if it needs to. I'm going to show you this.
Of injury to help fight infection. They are attracted there by chemical signals that are released by other cells of the immune system or by invading microbes. In this experiment, tiny amounts of chemoattractant are released from a micropipette. When neutrophils sense these compounds, they polarize and move towards the source. When the source of the chemoattractant is moved, the neutrophil immediately sends out a new protrusion, and its cell body reorients towards the new location.
All right, so that's neutrophil moving around. So neutrophils are part of your white blood cells. They are a white blood cell, and they can follow the scent of a bacteria, so it's really interesting. And the way that they move their bodies around, because these cells don't have muscles, what do they have to move around? It's the cytoskeleton. All right, so let's talk about 2 structures that are made from the cytoskeleton. Microtubules form cilia and flagella. So here is an image of cilia. Okay, so this is your cilia, and this is a flagellum. So cilia, maybe this looks like microvilli to you, but it's not. Cilia are very different than microvilli. Cilia are not meant to increase the surface area of the cell. They are meant to move stuff on top of the cell. So you see how this arrow is moving and pointing one direction. So these cilia are going to move and beat like oars. They are coordinated, and what they do is any mucus that's on top of the cells will get pushed in one direction. This happens in your respiratory system. So in the back of your throat and also down the larger tubes of your lungs called bronchi, there are your bronchi in your throat are coated with cells. I mean, the lining, sorry, I should say the lining of the cells is cells with cilia, and you're also producing mucus. So when you're breathing in, all the dust and debris and different kinds of things that are coming in with the air, they get trapped in the mucus, right? So the mucus will trap, because the mucus is sticky, it'll trap all this yucky stuff that you're breathing in. And then the cilia will keep moving that mucus up towards the back of your throat. So instead of landing deeper into the lungs, it gets trapped and then moved out of your lungs to protect your lungs from all that nasty stuff that you breathe in. And once it gets to the back of your throat, you swallow, anytime you swallow, you're swallowing a little bit of that mucus into your stomach and therefore destroying anything that harmful that may have been trapped in the passageway to your lungs. So cilia in your body, for humans, there's only cilia in that one place. So there's that one respiratory system. And then there's cilia in the female uterine tubes. So, and then there's a couple other places that are called stereocilia, a little bit different, but there's not a whole lot of places where we have that, but by and large, this is the most widely spread area of our body that has cilia, the respiratory system. For flagellum, right, the flagella is a very long whip-like tail, and the only cell that has this for the humans is the sperm. And of course, only men will produce the sperm, but that's the only cell that we have, flagella. And it's meant to obviously propel the sperm to help fertilize the egg. All right, our next one is centriole and centrosomes. This is an organelle that is meant to move the chromosomes around in mitosis and meiosis. This might look familiar to you. It should, because of Bio 3. And you can see that it's made of microtubules. So microtubules are called microtubules because, hey, they are a tube. And so anyways, these will, if you remember, the spindle fibers will just, these microtubules get longer and longer, and they help move the chromosomes. Okay, so just recognize this organelle. You can either call it a centriole pair, or you can call it a centrosome. All right. Let's talk about Roman numeral 3, intercellular attachments. So the cells of your body are attached and fixed to other cells, and they're also attached to connective tissue so that nothing is going to just flake off your body. I did mention the top layer of skin does flake off your body because those cells are dead and they're losing their attachments. But for all living cells, they are attached. And they do that with something called cell adhesion molecules. So cell adhesion molecules, for example, they would be these light blue structures here. So that's a cell adhesion molecule. There's another cell adhesion molecule on the other side, and they connect. And when they connect, they hold the cells together. Now, there's different kinds of ways that cells can be bound together. So I just have two for you here, letter B and letter C. One kind of junction between cells will leave a hole, right? This is called a gap junction. So there's a tunnel that actually is present between the two cells. So the two cells, the cytoplasm of one cell and the cytoplasm of the next cell, can actually be continuous through this little tunnel. So these gap junctions allow cells to communicate with each other and to almost share a cytoplasm. So they're going to be passageways where substances can pass through. They can coordinate actions because the cytoplasm is pretty much like one. So your heart muscles will have a lot of these gap junctions to coordinate the heartbeat, and your smooth muscle as well. And so that's what it is. So it's a gap gap junction. Now, there's the other kind of junction that we're talking about is called a tight junction. And tight junctions look different. So take a look at how it's pictured here. It's basically a zipper. So when you have tight junctions, the cell adhesion molecules do not form a pore. They tightly bind the membranes together. So if you wanted to have, I'm going to use the word airtight, but I'm using it loosely. It's basically a very tight junction, to use the name, so that molecules, you're creating like a barrier for molecules, and you're also creating this sheet of cells, because it's kind of like sewing all your cells together and you're creating a sheet. So for example, your skin, again, there's all these tight junctions that are holding your skin cells together. An example of this is if you go to the beach and you burn. My daughter actually had a really huge sunburn this summer, and she started peeling, and the skin that was coming off of her body, it was coming off in sheets because the tight junctions are there, and so it holds those cells together very, very tightly. All right, so where can you find these adhering junctions or tight junctions? You can find them on the sides of a cell. So if cells side by side, right on the sides, want to attach to each other very tightly, you'll find tight junctions there. You'll also find them anchoring to the basement membrane. Now, we haven't talked about the basement membrane yet, but the basement membrane, I just wanted to see what picture I had there, is going to be the bottom connective tissue that glues cells down to the surface. So this line that I drew, that would be a basement membrane. And the cells would have to have tight junctions attached to it. Okay, so it attaches side by side and also attaches to the bottom. One nice clinical example of this tight junction is celiac disease. And so let me show you what's going on here. So look at the tight junction between our two cells along the sides, and then notice that the tight junctions are open, right? They're not together. There's something wrong with our tight junction. So in celiac disease, they think that this might be the cause of Crohn's or celiac disease. This is gluten. So gluten in your intestines, if your intestines have junctions that are not functioning, the gluten can actually pass through the lining of your intestine and into underneath. So this is underneath your intestinal lining. Your immune system, right, would look at this and go like, what is that? This is a foreign object. This gluten should not be under your intestines. So the immune system will actually start to attack the gluten, and then when it does so, it mounts an immune response, and the immune response creates a lot of inflammation and problems here, which then go back and can destroy and be very, very harmful to the lining of your intestines, right? So in those with celiac disease, the junctions come apart, allowing large amounts of this indigestible gluten to seep underneath the tissues and incite the immune system. Treatments that reduced leakiness could potentially ease, but not only celiac disease, but other autoimmune disorders causing this unusually permeable intestine, right? So it shouldn't actually allow this gluten to permeate through, but it did. So that's an example of how tight junctions are very important. This is a very specific kind of tight junction. This is actually not mentioned in your outline, but this is a hemidesmosome. And this is the kind of tight junction that you see attaching to the basement membrane. Okay, so that's it. So it's a special kind of tight junction attaching to the basement membrane. All right, so I don't actually have a picture of mitosis and meiosis, but I do want to mention that you do need to know just the information that's in that box. So it's just a very basic overview of mitosis and meiosis. So I'm not going to go through all the steps, but for the first test, you will need to know this box, okay? And we're going to wrap up with some questions. So number one, and pause the video to make sure you can read it and try and decide the answer for yourself. So number one, some integral membrane proteins form gated channels that open or close to. So the answer should be letter A. Number 2, which organelle would be in abundance in immune cells that kill bacteria? The answer is letter B, lysosome. Products made in the ER would travel to the answer is C, Golgi. Should be Golgi apparatus. Number 4, functions of the plasma membrane include which of the following? Answer is A. And that's it. So that concludes our Chapter 2 lecture.